Current research into Alzheimer's immunology focuses on one piece of cellular machinery, the microglial NLRP3 inflammasome. This is the dominant view in the literature. On the left, we see the inflammasome acting as an engine of accumulation. The microglial cell expels particulate specks that cross-seed, clumping into the pathological amyloid plaques defining the disease. But a separate body of research observes a different behavior. On the right, the same cell releases catabolic enzymes. Instead of building plaques, it dissolves the protective extracellular lattice that normally shields nearby neurons. These two literatures, one tracking the proteinopathy, the other tracking the extracellular matrix, scarcely cite one another. They operate in separate scientific universes. Because these observations are isolated, researchers assume that building pathological protein and destroying a healthy protective matrix are two different, mutually exclusive microglial states. This creates a biological paradox. We have to explain how a single cell can simultaneously act as a failed janitor, allowing protein to build up, while operating as an aggressive demolition crew, tearing down the extracellular matrix. One singular activation event initiates two distinct destructions at once. The microglial cell enters a state where it loses the ability to clear debris, while it gains the drive to secrete matrix-degrading proteases. To understand this, we have to map the mechanism outward from the cellular root, rather than working backward from the disease endpoints. The inflammasome is a pleiotropic node. A single trigger yields a divided output. This tree diagram traces the activation from its root. The sequence begins when ambient amyloid beta interacts with receptors on the microglial surface, sending a signal inward to prime the cell. Inside the cell, an internal vesicle ruptures. This releases the enzyme cathepsin B, providing the secondary activating signal. Those components snap together at the core of the microglia, assembling the central NLRP3 inflammasome hub. This hub auto-activates the enzyme caspase-1. Caspase-1 drives multiple processes at once. It expels an ASC spec on one path, and it matures the cytokine interleukin-1 beta on the other. This single cellular node is the shared root feeding two distinct outward branches of pathology. One biological trigger at the sensor guarantees this divided multi-pathway attack on the brain. Following the left branch, or branch A, the expelled ASC spec exits the cell. In the extracellular space, it acts as a physical anchor, binding and cross-seeding ambient amyloid into dense plaques. Simultaneously, the matured interleukin-1 beta molecules travel upward to bind with nearby neurons. Inside the target neuron, this cytokine shifts the balance of internal kinases. The structural tau proteins become hyperphosphorylated, causing them to tangle and misfold. When we evaluate this specific proteinopathy pathway using a strict evidence grading rubric, the support is rigorous. Because it is supported by genetic deletion studies in mouse models and corroborated in human tissue, Branch A carries high Grade 1 and Grade 2 evidence. This mechanism is a validated amplifier of Alzheimer's proteinopathy, yet treating it as the entire disease ignores half of the inflammasome's output. Branch B tracks the other half of that divided output. Here, the same interleukin-1 beta induces a protease secretome. The cytokine induces the release of enzymes known as ADAM-TSS aggreganases. These aggreganases cleave the perineuronal net, the protective lattice that shields parvalbumin interneurons. The result is that the parvalbumin interneuron is stripped of its primary biophysical and oxidative shield. The evidence grading here reveals a contrast. The cytokine-to-protease mechanism is a Grade 1 fact in cartilage biology, but in the Alzheimer's brain it is Grade 3 or 4. It relies on molecular homology transferred from joint tissue rather than direct end-to-end proof in the brain. While theoretically sound, Branch B exposes an evidential gap in how we understand Alzheimer's progression. Analyzing these two branches in isolation has blinded the field to their interaction. The two outputs curve back inward, converging on a single neuronal target. Down branch A, the inflammasome actively drives tau hyperphosphorylation from the inside. Simultaneously, down branch B, the inflammasome removes the net from the outside. An intact perineuronal net is a physical barrier against the internalization of pathological tau. This creates a synchronized pincer on tau. One arm builds the toxic seed, while the other removes the shield meant to block it. Neither the amyloid-centric nor the matrix-centric fields can observe this coordinated attack, because their isolated models only account for half of the mechanism. The inflammasome is structurally designed so that whenever a tau seed is built, the neural defense against it is simultaneously dismantled. This structured ledger allows us to adjudicate the two pathways. We must separate the sheer weight of the evidence from the mechanism's proximity to actual cognitive decline. Branch A decisively wins on pure evidence weight, anchored by established genetic deletion studies. But when we move to the cognitive proximity metric, the dynamic shifts. Branch B acts directly on parvalbumin interneurons and the gamma-frequency timing of cortical circuits. The health of parvalbumin interneurons and robust gamma-frequency timing correlate tightly with actual cognitive collapse, more tightly than amyloid burden. Therefore, Branch B dominates the proximity metric. There is a significant difference between how well we know a mechanism and how much that mechanism actually drives the clinical disease. The most rigorously evidenced pathway may not be the one most responsible for the patient's collapse. This academic adjudication informs practical clinical strategy. Deciding in advance which of these two branches matters more is not necessary for therapeutic intervention. Because both of these destructions stem from the same central NLRP3 caspase-1 node, a single selective inhibitor interdicts both pathways simultaneously. Applying this inhibitor converts a philosophical debate into an empirical measurement. It tests if patient benefit tracks the physical proteinopathy, the matrix and network readouts, or both at once. The aggregative arm is what we best know. The proteolytic arm is what we most need to explore. But their shared biological root is the specific choke point that we can safely act on right now.